working for Matt Hurls and Jeff Barrett at the Sanger Institute. And today I'm going to talk to you about some work that we've done looking into the prevalence and architecture of dominant developmental disorders. And this is within the DDD study, Deciphering Developmental Disorders study. And this is a study within the UK, which is a collaboration between patients and their families, along with the UK National Health Service and the Wellcome Trust Sanger Institute. And the objective of the study is to both understand the genetic basis of developmental disorders and also to catalyse improved diagnosis of these disorders. In the first instance, this has been funded by both the Wellcome Trust, the Department of Health and the NIHR. And from 2016 onwards, this is going to be funded by the Wellcome Trust alone. So our strategy was that we recruited 13.5 families with affected individuals. All of the individuals that were recruited into our study had been previously screened by clinical microarray. So this is excluding a number of individuals who might have large copy number variants that might be contributing to their disorders. All of the patients were recruited through the regional genetic services within the UK and Ireland, and the recruiting clinical geneticists performed systematic clinical phenotyping of the patients. And this involved taking things like anthropometric data, but also looking at the phenotypes of the individuals and coding their phenotypes through the use of the human phenotype ontology. Initially, the study was was investigating these individuals through microarrays, but we've subsequently shifted to performing exome sequencing on these individuals for genome-wide genetic analysis. A key part of the study is that we've been feeding back likely genetic diagnoses, and that's we do that through to the recruiting geneticists, who then validate and pass on the variants to the families if they successfully validate. Today I'm going to be talking to you about a subset of the study with 4,300 individuals, all of whom have developmental disorders. These individuals have a relatively heterogeneous phenotypes, although the bulk of them, over 80%, have intellectual disabilities. Around about a quarter have experienced seizures, and I think around 10% have autism spectrum disorder. Since they're trios, we sequenced all the members of the trios, that is the child and both parents, and we did this in order to identify de novo mutations. We've taken a fairly liberal approach to identifying de novo mutations. Our intent was to identify a high sensitivity de novo set. And by doing this filtering, we identified 8,300 candidate coding de novo mutations, and that equates to around 1.95 de novos per individual. And this is slightly higher than what we expect the true rate is, but the intent was to capture a high sensitivity set, and then use validation to exclude genes that that exclude or include genes that pass genome-wide significance. Around nearly a quarter of our individuals have a likely pathogenic de novo mutation. And what this means is that these individuals have a de novo mutation within a gene that we know is dominantly associated with developmental disorders. As part of the study, we've been setting up a database, a curated database of DD, developmental disorder genes, which we call the DDG2P. And this contains around 1,300 genes, of which just over 400 have been identified as with a dominant or X-linked dominant mode of inheritance. And these pathogenic de novo mutations are the biggest diagnostic category in our cohort, with maybe slightly less than 10%. We expect slightly less than 10% will be due to recessive causes. Given these likely pathogenic de novo mutations and the phenotypic data collected from our collected on our individuals, we were interested in looking at factors that relate to whether or not they have pathogenic de novo. On the left, I've included odds ratios and betas for a number of binary and quantitative traits. I won't go through all of these particular phenotypes. I'll just highlight some of the ones that we found reasonably significant associations. For example, we found that females were much more likely to have a pathogenic de novo than males, as well as individuals who had an affected relative were significantly less likely to have a pathogenic mutation, along with individuals who either had self-reported consanguinity or where we had identified consanguinity from their genetic data alone. And this lower likelihood of pathogenic de novo mutation is probably due to these individuals having a higher chance of having a recessive disorder.
Hey, Jeremy, it's Dennis.
Yes.
One thing, you can use your mouse and point to things, so we see just as a point. Oh, yeah, perfect. Thank you.
Okay. Sorry. Yep, recessive, probably due to a recessive disorder here. Recessive disorders here. We also collected a number of developmental milestones, such as the age at when the children first spoke, or walked independently, or sat independently. Most of these didn't show a significant association, but the age at which children first spoke did show a reasonable association. And the reason why the age of first words probably showed an association, whereas the others didn't, is that parents are probably more likely to both note and accurately record the age at which their children first speak. The other association I'll point out is that we found a relationship between the length of autozygous regions within individuals and chance of having pathogenic mutation, and that's shown in the plot on that relationship is shown in the plot on the right, where individuals with longer autozygous regions are much less likely to have pathogenic mutations. Although I should note that even individuals who are roughly equivalent to being the offspring of first cousins still had around about a 6% chance of having a pathogenic de novo. One association we didn't observe was we didn't observe any association between parental age and having a pathogenic mutation. Although when we looked for rather than just having a pathogenic mutation, but having the absolute number of mutations, we found associations with both paternal and maternal age. So we found that children with older fathers have on average an extra 1.5 de novo mutations per year. And this paternal age effect has been reasonably well established by a number of other studies. But the maternal age effect has only been more recently shown in the last year or two. And what we found was that older mothers contribute on average an extra 0.9 extra de novo mutations per year. Moving on from this, from looking at the factors that relate to the likelihood of having pathogenic mutation, we also performed gene discovery. We attempted to identify genes associated with having de novo mutations in our cohort. So what we've done is a typical approach using the expected rates of background mutation within genes and testing for enrichment above those background rates of mutation, giving under a Poisson distribution. So what we've performed is kind of 2 tests per gene, one for looking solely at enrichment of truncating variants within the gene, and one looking for enrichment of protein altering variants within the gene, where we also combine that with a test to see how closely the closely missense variants cluster in proximity within the genes. And we think this increases our power for identifying genes with a gain of function mechanism. I say we did 4 tests per gene because we ran these two types of tests, both within our DD cohort alone, as also along with also running this in more of a meta-analysis. And this was including other cohorts that have also identified de novo mutations in disorders like similar to developmental disorders. And this is, I've got a plot on the right which shows the proportion of individuals who had a pathogenic de novo mutation that is, a likely pathogenic mutation that is a mutation in one of our dominant DD genes, broken down by different disorder types. So what we see was individuals with intellectual disability have quite a high chance of having a de novo mutation in one of these genes, and that's similar to our cohort, but also that individuals from epilepsy studies as well as autism spectrum disorder studies also have a high chance of having a de novo mutation in one of these genes. And so we included these cohorts in our study. But I should note that we excluded ASD individuals who were unlikely to have severe intellectual disability, because these had a lower proportion of lower likelihood of having a pathogenic mutation in one of our known genes. This slide shows a Manhattan plot of all of the genes for which we observed at least one de novo mutation within our cohort, or a meta-analysis cohort, maybe just our cohort. So we've got around, in this we've got 94 genes that exceed genome-wide significance, 80 of which had been previously established as being DD genes, and we've managed to identify another 14 novel genes that are associated with developmental disorders. And so we've reported the variants within these genes back to the families for them to be able to help with their clinical progress. With some of the known genes that we expected to come out on top do so, such as genes like ARID1B and Ankyrin D11, where we've got around 30 individuals each with de novo mutations in these genes in the 4,300 individuals. For some of the highest ranking genes, we've got sufficient individuals for whom we had collected high quality facial photographs that we were able to create these average composite facial images, and these are shown for the 6 most strongly associated genes at the top. These don't contain any information that can be used to identify any specific individual that's contributed to these photographs. But what we think these can help with is help to identify the specific facial dysmorphologies that might be associated with specific genes, such as the characteristic face for KMT2A. Most, and I guess ADMP also has a specific facial dysmorphology here. This slide shows a summary of the phenotypic data from the individuals with de novo mutations in our 14 novel genes. And these genes were relatively heterogeneous in terms of their phenotype. We broke down the human phenotype ontology terms that were collected for these individuals by a number of different organ systems. And so we found that some genes showed a predominance within, say, within the facial terms, whereas other genes showed a predominance within neurodevelopmental terms. Given the large number of, given the scale of our cohort and the large number of de novo mutations in our cohort and the large number of genes that we've been able to identify genome-wide significance, one thing we wanted to do was perform a comparison of the power of exome versus genome sequencing to capture genes at genome-wide significance. And for this, we sampled from our cohort, different numbers of trios, assuming a fixed budget and assuming a different relative cost of exome to genome sequencing. So obviously, with a higher genome sequencing cost, we would be able to sequence fewer individuals. And the grey bars here indicate the relative, the current relative cost of exome to genome sequencing. And for all but the highest relative costs, when the cost of exome sequencing approaches the cost of genome sequencing, exome sequencing is much better powered to identify genes at genome-wide significance, and probably also much better powered to identify pathogenic de novo mutations within individuals for a given cohort size. And this is even despite the allowing for an increased sensitivity of genome sequencing to identify de novo mutations. Another thing we did was we looked at the power of phenotypic stratification within our cohort. So I mentioned earlier that around a quarter of our cohort experiences seizures. So what we did was we took those individuals who subsetted to those individuals with seizures and ran the same kind of enrichment tests to see how many genes would exceed genome-wide significance. And we compared this to a set of known seizure genes. And what we found was that within our complete cohort, we identified more known seizure genes at genome-wide significance than if we subsetted down to just the subset with seizures. And this highlights the power of large studies, the power of large studies compared to what you would get if you had a smaller subset. Similar to, in contrast, when we did another kind of analysis where we matched on sample size. So we again sampled from our cohort, from the complete cohort, and also sampled from the set of individuals with seizures, and tested to see how many genes we found we would get that would exceed genome-wide significance. And doing this across a number of different sample sizes, we found that the samples from the seizure subset contained more genes at genome-wide significance. And this is probably due to the fact that these seizure individuals are much more enriched for de novo mutations within these known seizure genes, and therefore we're better able to recapture those genes. And this highlights the benefit of doing a phenotypically homogeneous cohort. Earlier on in our study, we had performed some power estimates to estimate what proportion of dominant developmental disorder genes we would be able to capture across a range of cohort sizes. And from this, we had estimated that when we would have around 4,000 individuals, we would be able to we would have about 50% power to capture dominant DD genes. And we looked at the way we looked into this in this cohort to measure that was we assessed how many of the haploinsufficient genes with a neurodevelopmental phenotype exceeded genome-wide significance. And what we found was that 47% of these genes exceeded genome-wide significance. But we also expected that some of the haploinsufficient neurodevelopmental genes would be depleted within our cohort because individuals with defects in these genes will be screened out before they are recruited into our cohort. So we had clinicians rate the clinical recognizability of these haploinsufficient neurodevelopmental genes, and then we plotted the enrichment of de novo mutations within genes in each of these recognisability classes. And what we found was, as expected, genes that had a clinically distinctive phenotype were relatively depleted compared to the other recognisability classes. But we also observed that there were some genes that had a low clinical recognisability, but had a sufficiently high expected mutation rate. But where we also didn't observe any de novo mutations in our cohort, and we wanted to try and test whether or not these might be due to the fact that these aren't particularly good developmental disorder genes. So what we set up was a comparison of two models, one where we assumed a mutation rate that the gene wasn't a haploinsufficient gene, and one where we assumed that the mutation rate was for a haploinsufficient gene, given the enrichment of de novo mutations within the genes with the various recognisability classes. And we estimated the likelihood of observing getting our observed number of truncated variants under these two models, and ranked the genes by delta AIC between the two models. And I guess I don't want to mention too many of the genes, but highlight one particular gene, Exome 2, which had a very high mutation rate, but we didn't observe any de novo mutations. And this came out towards the top of the table, and we think this is a good candidate for not being a haploinsufficient neurodevelopmental gene, but one that's just piggybacked off a related gene, which is a good candidate. Moving on from that, we also looked into the excess of de novo mutations within our cohort. Before I mentioned that we had looked at the sensitivity, we'd tried to capture a high sensitivity set of de novo mutations. And in order to accurately measure the excess of de novo mutations, what we did was we recalibrated our de novo mutations so that we had the same number of observed synonymous mutations as we expected. And that was this selection was done using a quality threshold that we've got for our de novo mutations that estimates the posterior probability of being a de novo mutation. And this is down the bottom here, I've got a kind of a plot of sensitivity versus PBV across the various thresholds that we tested. And using the threshold we selected, it looks like we're doing relatively well on the sensitivity versus PBV. Observe an excess of 576 truncating variants and an excess of 1220 missense-like variants, or around about 42% of our cohort have a pathogenic de novo mutation. For the missense variants, what we wanted to try and estimate the proportion of those that had a loss of function mechanism, and we've done this in two ways. And right here, I've given the first of those ways. So given the 1220 missense mutations and the proportion of truncating and missense variants within haploinsufficient genes, so these missense variants have these 325 missense variants have a loss of function mechanism. That gives us the proportion of loss of function variants that will have a missense classification. And from this, we can estimate the number of our missense that have a loss of function mechanism, and our excess have a loss of function mechanism versus 41% with an altered function mechanism.
Jeremy, that's Dennis. One question. This model assumes that a loss of function gene, so that there's for each gene only one mechanism of disease, right?
Yeah.
So for example, it's not considering the option that you can have a loss of function variant, which leads to like a haploinsufficient reason for disease, but where a missense mutations can lead to, for example, a gain of function or a dominant negative effect, which also leads to a similar-ish phenotype, right?
Yes.
Okay.
Sorry, do you want me to move on? Sorry, I wasn't sure what the question was.
Sure.
Right. So we also looked into the breakdown of different functional categories by the PLI-based loss of function intolerance score. And as expected, we found that there was no, so this is based on, this shows the enrichment of observed over expected in genes by different PLI quantiles. And as expected, there was no in excess of synonymous mutations within high PLI genes, but we did observe a high enrichment of truncating variants within the highest PLI quantiles. And as well, we also observed an increase in the missense in high PLI scores. Okay, as before I mentioned that we estimated the proportion of missense with a loss of function mechanism, and that was by doing it one way. And this is, now I'm going to mention the other way that we've estimated this, and this was through looking at the distribution of missense variants amongst all genes by constraint quantile, and that's assuming our true model of missense variants. And we also looked at the distribution of missense variants in haploinsufficient genes, as well as missense variants in non-haploinsufficient genes. So we're assuming that this purple distribution is a mixture of these two other distributions. And what we did was we took a range of mixing proportions and calculated the goodness of fit to this observed distribution, and just selected the proportion with the optimal goodness of fit. And that's shown in this figure here, where we've also estimated the kind of the confidence intervals around that estimate. And from this, we've calculated that 57% have a loss of function mechanism, and this is very similar to our earlier estimate. Finally, we also looked into the prevalence of developmental disorders due to de novo mutations, and we've done this by calculating the cumulative loss of function mutation rate in haploinsufficient dominant DD genes, and then taking the prevalence of loss of function mutations within these genes, and we scaled this by the proportion of our excess to pathogenic loss of function de novos in dominant DD genes, and we also included an additional de novo CNV burden because these individuals had been screened out from being included in our cohort. And from this, we've estimated the prevalence at the average prevalence at around 0.34%, or around 1 in 295 births. So this is an estimate for the general population, given the average age of the individuals in our cohort. And what we did was we scaled across to different parental ages, given our age-specific parental mutation rates. And that's shown in the plot on the right here, where, and I'll just highlight that the two extreme ends of this range, where parents who are age 20 will have a risk of around 1 in 450 of having a child with a developmental disorders due to a de novo mutation, whereas parents who are age 45 will have a risk of closer to 1 in 200. We also tried to highlight the nature of the problem by estimating the annual prevalence of developmental disorders due to de novo mutations. And this, we did this by looking at the population of countries around the world, their fertility rates, and the age of first births, in order to calculate the average age of first birth at around 22 and a half years, and an average fertility rate of around 2.5 children per mother. And given an average interpregnancy interval of around 29 months, we've calculated the mean age globally at child's birth at around 26 and a half years. Given our age-specific prevalence at this age of around 0.29% and a global birth rate of 18.6 individuals per live birth, 18.6 live births per 1,000 individuals, and a total population of 7.4 billion individuals, we've calculated that there are nearly 400,000 births with developmental disorders due to de novo mutation per year. And we estimated that this is a greater burden than those due to trisomy 21 than the 3 common trisomies. Okay, I'd like to finish by acknowledging all the clinicians who recruited and phenotyped individuals into our study, as well as all of the large number of individuals who worked internally in Sanger to both process and analyze the data involved in the study. Thank you for listening.